Welcome to Clue's review of biochemistry in the cell cycle. Let's get started. The cell cycle is a process by which one parent eukaryotic cell divides into two daughter cells. The cell cycle is made up of 3 to 4 major phases depending on which phases you count as their own. The G0 or G0 phase is when the cell is halted in replication, typically awaiting enough growth for replication to proceed, so no division is occurring in this stage. Interphase consists of G1 or growth 1, S or synthesis, G2 or growth 2, by which the cells double their organelles and then replicate the chromosomes so that each daughter cell has the ability to grow and function on its own. The third stage is mitosis, which is the cycle of allocating one full set of chromosomes to each cell through a process known as PMAT or PPMAT. The last stage is cytokinesis, in which a complete set of chromosomes and organelles are on either end of the cell, and the cell may now divide its cytosol into two daughter cells. One acronym that I have used in the past to remember the phases of the cell cycle is Great Singers Get More Clout, which leaves out G0, but the G in Great refers to G1, S in Singer refers to synthesis, G in Get refers to G2, M in More refers to mitosis, and C in Clout refers to cytokinesis. Now we'll move on to reviewing the P or PPMAT of the process by which the chromosomes are divided. First off, the P in PMAT, or the first P, refers to prophase. The second P refers to prometaphase. The M refers to metaphase, the A refers to anaphase, and the T refers to telophase. So, in prophase, centrioles form in the cell and create projections of microtubule, with the nucleus beginning to disappear so that the centrioles can attach to the chromosome and begin to pull them. Prometaphase is more commonly known to pull the chromosomes so they begin aligning where they are needed. In metaphase, the chromosomes are aligned in the middle of the cell, which helps organize the chromosomes so that each daughter cell ends up with one copy of each chromosome. Anaphase is the process by which the chromosomes are pulled to either end of the cell, with the cleavage furrow beginning to form at the equator. Note that the cleavage furrow is the division in the cell that allows for one cell to become two. In telophase, the beginning of two separate cells forms, with the nucleus reappearing, and you can still see the cleavage furrow. And then in cytokinesis, the complete division of one cell into two with a full set of organelles and chromosomes occurs. The way in which I remember mitosis is through PMAT. And in some classes, you may not have to really know about the prophase or the prometaphase, in which case you would just combine the prophase and the prometaphase together. But in this case, I taught you about both. So in PPMAT, the first P stands for prophase, which is pull. The second P stands for prometaphase, which is pull more. The M stands for metaphase, which is middle. The A stands for anaphase, signifying apart. And the T stands for telophase, which is two. And the C in cytokinesis can stand for complete, if that's helpful for you as well. Lastly, in today's lesson, I'm going to give you a quick 3-question quiz over the material discussed. Be sure to pause the video to take time to think about your answers. 1. What phase of the cell cycle contains a checkpoint to ensure proper chromosome replication? 2. In which phase or process does one parent with double the organelles and chromosomes divide its cytosol to become two daughter cells? 3. During which phase would you find one full set of chromosomes at either pole of the cell? Everyone should be sure to check out Clue's website, our Facebook page, Instagram, and TikTok for more videos just like this.